Bismillahirrahmanirrahim. Assalamu alaikum. This short session is being recorded just to understand biostatistics and its application. First of all, we need to learn what is biostatistics. As you all know, bio means life and statistics is facts and figures. So all the facts and figures related to life sciences is biostatistics. Or in other concept, this is the application of statistical and mathematical tools to solve biological data or to analyze biological data. Biostatistics has two branches. First is descriptive biostatistics and other is inferential biostatistics. Descriptive biostatistics means in which we collect the data and describe it. We see the pattern, we see the distribution, and we just see the trends of the data. And in inferential statistics, we test the hypothesis, we draw the inference, we draw the conclusion for population on the basis of sampled information. So for descriptive statistics, when we collect data, there are mainly three important ways to present our data. One is table, other is graph, and is single measure that is usually called central tendency. In graphs, there are many options, but you need to know about the application of those graphs according to the type of data. So first of all, we will discuss about the types of data. There are mainly two types of data. One is categorical and other is quantitative data. Categorical data is type of data that in which the answer is categorized according to the different characteristics like gender, like blood groups, like anemic status, like status of diabetes mellitus, and in quantitative data, the response is always in number. If someone asks you what is your age, you will tell the number. What is level of HB, you will tell the number. When we tell the numbers, it is quantitative data, and when we describe the characteristics of any variable, it is categorical data. Categorical data can be measured on ordinal and nominal scale. Nominal scale is derived from the word name and that describe the characteristics only, that gives the name to different characteristics. Like if you ask someone that which kind of disease you have, he or she will tell you the name of disease. If you see if someone is male or female, this male and female is actually the collective name to the group having similar characteristics. When these nominal data have natural order, have ranks, it is called ordinal data. We need to understand the difference between nominal and ordinal. When it is distinguished by its name, it's nominal. When it is distinguished by its name and there are natural order, ranks, grades, it is ordinal data. All the statistical analysis plan should be according to the type of data and objective of the study. So quantitative data is measured for ratio and interval scale. Interval scale is a type of measurement scale for quantitative data in which zero comes as an origin and zero means presence. There are other characteristics too, but for your convenience, I will focus on the on the uh, on the concept that zero is present and when zero is origin. When data can be collected as zero, that gives presence, like temperature. If today's temperature is zero, it means it is present. And for ratio data, where data is absent for zero. So nominal and ordinal are for categorical data. Ratio and interval are for quantitative data. There are other few concepts like variables and constants. Variable is a characteristics that varies from individual to individual and that has variable chance for the data. Like if you ask someone what is your age, what is your height, what is your salary, what is your income, what is your grade or CGPA in your exam, there are chances that everyone will answer in different way. If you ask someone and there are no chance of variation, it is constant. There are few important types of variables like independent variables and dependent variables. Independent variable is a type of variable in which we see the effect of some characteristics on another variable or characteristics that is called dependent variable. Dependent variable is always modified in the presence of variables that are called independent variable. And if the relationship is confused with independent and dependent variable, and if there is another third variable that affect these both, it is called confounding variable. There are many other types of variable, but we will focus on independent, dependent, and confounding variable. When we collect data for quantitative, for categorical, for nominal ordinal, for interval and ratio, and on these variables we need to present this data. There is one more concept regarding quantitative data. It can be of discrete or continuous. Discrete data is type of quantitative data that varies jumps to jumps, like number of brothers and sisters we have, like pulse rate, like heart rate. They are not measured in decimals usually. And if we can measure data in decimals or that comes through each and every point between two numbers, it is continuous data, like ESR level, like CRP level, like creatinine level, and other parameters in hematological profile that we usually collect. So when we collect data on quantitative or categorical data, we need to present it graphically. There are common graphs that are used is pie chart. Pie chart is used for categorical data when there are less categories. Bar charts, they are used for discrete data or categorical data or ordinal data that have more categories. For continuous data, we use histogram when we need to see the behavior of data. If we want to see the outlier in the data, we use box plot. If we want to see the relationship between independent and dependent quantitative data, we apply scatter plot. If we want to check the normality of data, we apply PP plot and QQ plot. These graphical presentation will just give you an idea about the trend and nature of the data behavior or distribution. If you want to test them, for example, if you want to check the normality, graph will not give you the p-value or evidence about the normality of data. For that, you need to apply some statistical tests. We can make one-way table for categorical data, two-way table when we need to present more than one variables like smoking status versus gender. We calculate mean and standard deviation for quantitative data when there is no outlier and data has no abnormal measures. We apply median and interquartile range when data is not normal. Data is not normal mean data is skewed. Data can be positively skewed if you plot the data or if you make histogram of the data. If the right tail is prolonged, it means data is positively skewed. If it is negatively skewed, the left tail is prolonged. In positive skewed, mean is greater than median is greater than mode, and in a negative skewed, mean is less than median, they both are less than mode. Standard deviation and interquartile range are actually the dispersion of data that tells us how the values are far away from their standard values. For mean, we use standard deviation, and for median, we use interquartile range. Median and interquartile range are usually presented in box plot that helps us to see the outlier in a data. So this was all about commonly used descriptive statistics. In the next session, we will cover the major concepts of inferential statistics.